biological molecule, okay? I mean the organic compound. Okay, so you already know that we have 4 types of organic compound, right? Which is those, okay, which is the carbohydrates, proteins, nucleic acids, and then one more is the lipids. Those are the organic compound, okay, which is they contain the carbons, hydrogens bond. And then we already discussed about the type of the carbohydrates, right? We have 3 types, which is the monosaccharide, disaccharide, and the polysaccharide. Okay, so we already know about how monosaccharide, like the glucose, how they can join together. I mean, how they can combine together by the bonds, so they can become the disaccharide or polysaccharide, which is a much more complex molecule, like the glycogen, starch, and cellulose. Okay, so how they able to combine together to form a disaccharide or a polysaccharide is by the condensation. You can see condensations, okay, which is you need to join the bond together. Join these 2 molecules together by a bond. Okay, and also you also can split them. Means that you can split the disaccharide become the monosaccharide again. So how you able to split them? The process that you separate the molecule become their monomer. The process is known as what? Involved in the addition of water. Maybe you can have a look. This one. From the sucrose become the glucose and fructose. The process is known as the Yes, it's hydrolysis, right? Because you are required the additions of the 1 water molecule added to the sucrose. So this water molecule, right, it will split the bond. You can see, right, there's a glycosidic bonds combining these 2 molecules together. So they'll break down this bond, so they'll become their separate unit of the monomer. So this hydrolysis. You want to separate them, means you use the water to split them. So it's known as hydrolysis. If you want to combine them together, they're known as condensations. That's all. Okay, and then we talk about the fats, right? So you already know that if we want to have 1 molecule of triglyceride, that means this triglyceride is being composed of 1 glycerol and 3 fatty acids. See, those are the 3 fatty acids, okay? And then fatty acids, right? I think I tell you, right? Fatty acid, we have 2 types, saturated and unsaturated fatty acid. Did I tell you before? Saturated fatty acid. Yeah, saturated fatty acid and unsaturated fatty acid. So just very simply, if you can see all are comprised of single bond like this, I can see, right? All are comprised of this single bond. How can I know it's single bond? Because I see from here, see, only single bond. Only single bond. Where is my pen? You can see, right? Only single bond. So this one we call it saturated fatty acids. If you have 1 molecule which is double bond, maybe you become like this. Okay, that means it is a unsaturated fatty acid. So you just observe how is the bond formation. And then we talk about the proteins. Okay, so protein is being made from the amino acids. It's being made from the amino acids. Okay, so here is the amino acids. And then we're able to join those amino acids together, become a polypeptide chain, and then we'll produce a final product, which is a protein. So Chloe, can you give me example of the protein product that was being produced in our body? Okay, take for example, something like the enzyme. You heard about enzyme, right? Enzyme. Yeah, and also hormones. Okay, the hormones. Yeah, enzyme, hormones, and the antibody. Okay, so those are protein product. Example of protein product which is produced by our body cell. For example, enzyme will be produced by what kind of the organs? What organs will produce enzyme? For example, stomach, right? Stomach able to produce an enzyme, which is a pepsin. to help in the protein digestion. Pancreas also, pancreas, they can produce the enzyme. Those organs, they will produce the enzyme. Then how about hormones? Okay, you will learn later, okay, which is the organs, they will produce the hormone known as the endocrine gland. Pancreas is one of the endocrine gland. Okay, other glands, they might be the, for example, in the male will be the testis, in the female like us will be the ovum. Okay, it's not ovum, sorry, it's an ovary, right? It's an ovary. Ovary is the organs, they help to produce the hormones, right? I think you heard of this hormone before, right? Estrogens. Yeah, estrogen. Okay, the hormone which they make you become appear look like a female, estrogen, because they give you the secondary sexual characteristic. And also any kind of hormone like the thyroid gland. So those are the products which made from the proteins, okay, which is they are composed of the amino acids. You can see, so this is amino acids. So we talk about protein, right? We talk about these 4 level organizations of proteins, both from the primary structures, which is your amino acid is being linked together by a simple peptide bond. Okay, see, you can see, right? This is a peptide bond, which is here. This is a peptide bond, this one. Oh. Okay. Here is a peptide bond. Okay, peptide bond. They're able to link the amino acid together. So these peptide bonds, right? They will join the amino acid together, and then they'll become a polypeptide peptide bond. Okay, Chloe, peptide bond is a very, very strong bond. It can only being break down by what? By the protease. By the protease, right? Protease enzyme. So you can see that this peptide bond, they will join the amino acid together, and then they will form a polypeptide chain like this. So this is a polypeptide chain, okay? Polypeptide chain. Polypeptide chain. Then after that, the polypeptide chains, they can continue form the bond to each other, then will become a specific structure. So they are being joined by the hydrogen bond. So you can see now they will produce a secondary structure. So after that secondary structure, they will further fold together. They will become tertiary structure, which is they will become a ball-like shape, we know as globular. So if you have more than 2, 3 polypeptide chains, they join together, you form a quaternary structure. So example of quaternary structure will be the hemoglobin, okay, your red blood cell. Your red blood cell is made from the proteins. It's made from the hemoglobin proteins. Okay. Proteins. Okay, so the most important aspect for the proteins will be their shape. Because the shape of proteins will determine the function of the protein. As simple as that. So if you have any factors, they induce a change in the protein shape, you will be able to affect the protein functions. Means that if there's any change in the shape of proteins, it will cause them become a non-functional protein already. Because the protein product, right, they are unable to bind to their substrate. Because their shape already changed, being altered already. So what are the factors able to alter the shape of the proteins? Can you give me like one of factor that can change the shape of the proteins? Something like initially they become like this, right? From like this, right? They will change their shape, right? Become something like this. You see, they totally lost their 3-dimensional shape already. So there will be 2 factors, right? We call as temperature and the pH. So these 2 factors, right? They can able to change the structure of the proteins. First is temperature, another one with the pH. Okay, so this will bring to our chapter for today, which is the enzyme. Okay, so later we'll talk about how enzyme structure being affected by those 2 factors. temperature or the pH. Means that if the temperature become very high, right? Too high temperature. So you heard about the kinetic energy, right? Kinetic energy.
Yeah.
Okay, so you tell me, whenever the temperature increase, how about the kinetic energy? Will they increase or they will decrease? When the temperature increase, the kinetic energy will increase or decrease?
Increase.
Yes, it's increase. Yeah. So always remember that kinetic energies have a positive correlation with the temperature. Means that if you are increasing your temperature, right, you're actually increasing the kinetic energy of the molecule. Okay, you're actually increasing the kinetic energy of your enzyme and the substrate molecule. Means that they can vibrate faster. Okay, they can vibrate faster. But you can see that if the temperature become too high already, okay, too extreme temperature, so you're able to destroy the structure of enzyme. Okay, why? It's because that the kinetic of the enzyme molecule become increased rapidly already, too rapidly, until you break the bond. Until this high temperature, right, they're able to break the bond holding the enzyme. So that's why there's no bond holding the enzyme structure already. So the enzyme will become like this, okay? But they lost their specific shape. Okay, can you get me, Chloe?
Yeah.
Yeah, so means that the high temperature, they break the bond holding the enzyme structure. Okay, so that's why our protein, right, is very sensitive to any change in the temperature or any pH. Means that for the pH, right, pH means that the pH cannot be too acidic or too alkaline. They must be at their optimum point of the pH. Okay, every enzyme have their optimum pH. Okay, this one you will learn more later. Okay, that's all. So just remember that proteins structure will determine the protein function. So we need to try our best to maintain the protein structure. Okay, so yeah. So that's why if you're able to maintain the shape of protein structure, right, you can allow like the antibody, right? Antibody, they're able to bind to the antigens, and the enzyme able to bind to substrate because they are complementary to each other. So, and also the protein structure, right, is being determined by the sequence of the amino acids. Okay, Chloe, maybe you can have a guess. Do you know how many type of the amino acid in our body? I mean, how much of the amino acid contained in our body? Is about 20 type. We have about 20 type amino acid in our body. Okay, 20 type amino acids. So that means that these 20 type amino acid, they can be joined together in the different sequence. Right? So this different sequence of amino acids, they will contribute to the different structure of the proteins. So that's why contribute to the different function of protein. That's all. Okay, now we talk about food test. Okay, now we talk about food test. Food test is just a simple test which they're able to detect the nutrient contents in the foods. Okay, for example, the food nutrient molecule, right? When we talk about macronutrients. What's meant by macronutrients? Macronutrients means it's our body requires those nutrients in a very, very large amount. So example of macronutrients in our body will be the carbohydrates, for example. Carbohydrates will be the proteins, and then there will be the fats. Those are example of macroproteins will be required by our body. Okay, so the food, right? The foods inside, they also contain the carbohydrates, protein, and fat. Then the question is, how we able to detect the concentration of the nutrient molecule, or we can detect the amount of the nutrient molecule inside the food sample. So that's why we come up with the food test. So now we talk about the glucose test. Glucose, okay. So because that glucose, right, is being categorized into two types: reducing sugar and non-reducing sugar. I think this is your first time heard it, right? So what's mean by reducing or non-reducing sugar? So it just means that reducing sugar, right? When they test this food sample contains reducing sugars mixed with the benzylic solutions, and then you heat up this benzylic solution about 60 to 70 degrees Celsius for about 5 minutes only. Okay, not until boiling point, but it just heat up for a bit, for some time until they reach 60 to 70 degrees Celsius. So after that, you can observe there's change in the benzylic solutions. So initially, the benzylic solution will be, you can see, right, Chloe? This is the initial color of benzylic solution. So what is the color, Collie? What is the color?
Blue.
Blue, yeah, it's a blue. So why is the blue? It's because inside they contain the copper 2 plus. Okay, this one. They contain this copper ions. This one. So this copper 2 ion, right? Naturally, they will give this solution become blue color. So pretty, right?
Yeah.
Yes. Okay. So what will happen is, if this food sample they contain the reducing sugar, they can able to change the color from the blue color become green color, become orange color, then finally become brick red precipitate. This is the final color we want to get, brick red precipitate. Okay, so it means that if the food sample, right, and then this color change, this color change depend on the concentration of the reduced sugar. So if you have a trace amount of reduced sugar, it means that you just have very, very low amount of reduced sugar, not too high. So that means the color will change from blue color become green. But if you have a moderate amount of reduced sugars, the color will change from green become orange. But if you have a high amount of the reduced sugar, it will change become brick red precipitate. So this brick red precipitate means that they have a very large amount of reduced sugar. So what's meant by reduced sugar? Reduced sugar means that they're able to change the color of the blended solution, which is initially from blue color either become green, orange, and brick red precipitate. Depend on the concentrations. Depend on how high is the concentrations. Understand?
Yeah.
Okay, so this is a bit chemistry. Chloe, you learned about chemistry before, right? Or
Yeah.
now you are learning?
Yeah.
Okay, you are learning now, right? So Chloe, in the chemistry, right, I'm not sure, have you heard about reductions, oxidations? Have you heard about this stuff before? This one, reduction, oxidation. So Chloe, what's mean by reductions? Reductions means that either these atoms, right, they will accept the hydrogens, okay? Or they will accept the electron. Or they will donate the oxygens. So this we call as a reductions. So what's meant by oxidations? Oxidations means that these atoms itself, they will accept the oxygens. Accept the oxygens. Okay, so when they accept the oxygen, right, so they will remove the hydrogens, or they remove the electron away. Okay, remove the hydrogens, or they will remove the electron. So you can see reductions and oxidations. So what happened is, okay, when you mix this copper 2 plus with the blended solutions, right? I mean, when you mix these blended solutions which contain the copper 2 plus with the reducing sugar. So what we call that reducing sugar, we know it's a reducing sugar is because that it contains, we call it as a 3 ketones or aldehyde groups. 3 ketones or aldehyde groups. Okay, this is very new term for you. Okay, so just know that, okay, ketone is just to describe the groups. So like this, right? If you see any molecule, right, they are something like this. So that means it's ketone. This is ketone. Okay, but if you see any of the molecule, they become something like this with a H. That means it's aldehyde. Okay, see, they have COH. Ketone means CO. Aldehyde. So you can see, right, what happened is on the oxygens, right? Oxygens, you have the electron group on the oxygens. I mean, you have the free electron on the oxygens. You have like this. Okay, so yeah, we talked about chemistry already. This is about chemistry. Okay, I just let you know. Yeah, see, the oxygen, right? Oxygen, they have about 8 electrons. I think you learned before, right? Oxygen, they have about 8 electrons. So they have this free electron group. So what will happen is they able to reduce. Okay, so what's mean by reducing sugar? Reduce sugar means that they're able to reduce other. For example, they're able to reduce this copper 2 plus become copper 1 plus. Like this. So from copper 2 plus, they'll become copper plus. Okay, how they're able to do that is because they can allow this copper 2 plus, right, to do what? To accept the electron. Okay, so it becomes something like this. Copper 2 plus, right? Copper 2 plus, one they accept the electron, they become what? Copper plus. Okay, so where they can get electron? From here. Okay, from here, from this free ketone or free aldehyde group, they have the extra electron group, right? So they can donate electron to the copper 2 plus. So that's why copper 2 plus become copper plus, right? Actually, it's a process of the reductions. Okay, it's a further reduction. This is a reduction process. So reduction process means what? Means that they are actually reduced. You know, this reducing sugar, right? This is reducing sugar. Reducing sugar. Oh. Reducing sugar. Sorry, reducing sugar. They can reduce other. So how they can reduce other means that they can cause other to undergo reductions, but itself, right? Itself will be undergo the oxidations. Okay, itself will undergo the oxidations. Means that they undergo oxidation. So this copper 2 plus, they undergo reduction, become copper plus. And then this reducing sugar, right? They undergo oxidations. So now they become what? They become like this. Okay. So you can see they will addition of the oxygens here. So from like this become addition oxygens. That means they undergo oxidations. Yeah. Okay. They undergo oxidation at here. Oxidation. So Chloe, do you know what's meant by reducing sugar? Reduce sugar means that they reduce other, but itself undergo oxidations. So that's meant by reducing sugar. Okay, are you clear with it?
Yeah.
Okay. So it's like that. So reduced sugar, right, includes glucose, fructose, lactose, maltose, together. So many means that all those sugars able to change the Benzyl solutions from blue color become other kind of color. Okay, become brick red precipitate. Only the sucrose. Sucrose is not reduced sugar. Means that if you add if you add the sucrose together with the Benzyl solution, what will happen? If you add the sucrose with the Benzyl solutions, will the Benzyl solution change their color?
No.
Yes, no. So that's why they will just remain blue in the Benzyl solutions. Okay. The reason is because sucrose, right? Sucrose, the molecule is a bit different. They don't have the free ketone or free aldehyde group. So that means that they cannot donate their electron. Okay. That's all. So you just need to know that sucrose is the one non-reducing sugar. That's all. So this is an example of the Benzyl solutions. So you can see how to do it. Sometimes the IGCSE exam they will ask you about the Benzyl solutions, which is initially you need to add these Benzyl solutions into the test tube, right? So inside the test tube we contain the food sample already. So you just heat up the food sample with the Bunsen burner about 5 minutes until they get about 60 to 70 degrees Celsius, and then you can see this color change. That's all. And then now we talk about proteins, right? Proteins, which is protein test for the foods. So if any food sample, they contain the proteins, you can test them with the Biuret solution. This one, Biuret solution. So what happened is, Biuret solutions initially is also, originally is blue color. So if there's a presence of protein, right, the blue color become purple or violet. So they become purple like this. If they contain protein. If they don't contain protein, right, they will just remain as blue color. So this is Biuret solution. Okay. And then now we talk about the common test, starch. It's not starch, it's called iodine test, sorry. Iodine test, okay? So you can see what happened to this biscuit. Okay, so Chloe, you can describe how's the color change? From what color become what color?
From red.
Red.
It becomes black.
Yes. Okay, you can say that. From the reddish brown, you can say yellow brown. Yeah, so they become totally this is called a blue black. See, blue black, because it doesn't totally look like black color, but it's just like blue black, very, very dark blue black color. So if the iodine test, right, if they change the color from the yellow brown, from the brown color become the blue black color, means that this food, right, there's a presence of starch inside. Only the starch. Okay, so for example, if you put this iodine on the potato, they will also change their color. So you also the blue black color on the surface of the potato, because inside the potato they contain a lot of starch. Okay, starch. So starch are usually found inside the plants. Okay, so Chloe, why the plant they contain a lot of starch? It's because that plant, right, they will do what? Plant, they will undergo the photosynthesis, right? Photosynthesis, which is they will absorb this carbon dioxide and the water, and then they'll become what? They become the glucose, right? C6H12O6, which is the glucose, and then release the oxygen, O2, or what happened? O2. This one, O2. So during photosynthesis, right, plant will produce glucose, correct? So plant will produce the glucose at where? Okay, so Chloe, photosynthesis is being conducted at the which part of the plant? It occurs at the leaf part, stem, or roots. Choose either one. Where is the site of photosynthesis?
Leaf.
Yes, it's a leaf, right? So here is in the leaf. And also, only the leaf contain the chloroplast. There's only chloroplast found inside the leaf. Okay, there's only chloroplast found inside the leaf. There's no chloroplast exists in the stem or in the roots. Yeah. So that's why during photosynthesis, right, inside the leaf of plant, they will produce a glucose. So after the plant, they will produce a glucose. So what happened is they will convert this glucose become other kind of compound. Means that maybe they can use our glucose for respiration, or they can convert the glucose become sucrose. And then, or they can become starch. Like this. Okay, understand? Means that they can convert this glucose become another form of the nutrient molecule, and then they store inside the plants. Just like, for example, in our in human body, right? In our body, our liver also able to convert the excess glucose in the blood become the glycogen stored inside the liver. So we're able to convert the glucose from the blood, right? Become glycogen in the liver. Understand?
Yeah.
Yeah, so it's a kind of, they call it starch, right? So why starch is being discovered in the plants? In the plant cell, because the starch, right, is kind of energy storage compound of the plant, okay? Yeah, it's a kind of energy storage compound of plant. Maybe I can show something. Oh, what happened to my laptop? Okay, starch, right? You can see starch. It's the energy storage compound of the plant. See? Okay, starch is a primary energy storage compound in the plant. So it's just like the insoluble or the osmotic inert form of glucose. So during photosynthesis, right, plant, they can convert excess glucose become starch and then store inside the chloroplast. Okay, so where you can find the starch? Starch are found inside the chloroplast, right? Chloroplast is a kind of organelles. Okay, it's an organelle found inside the plant cell. Not all plant cells contain chloroplast. Okay, I think you know, right? Not all plant cells contain chloroplast.
Yes. Yeah.
Only the 3 plant cells contain chloroplast. So what are the 3 plant cells? Is the mesophyll cell. Okay, in the leaf, right? In the leaf, we have 3 types of plant cells contain chloroplast, which is the pellicet and spongy mesophyll cell. This one, pellicet, and the spongy mesophyll cell. Oh, and then we have the gut cell. It's a gut cell. Yes, that's it. Okay, that's all. So another thing is, because starch, right? Okay, Chloe, starch is a polysaccharide or is a monosaccharide or is disaccharide? Which one? Starch is very large and very complex, so it belongs to which saccharide?
Polysaccharide.
Yes, very well done, well done. Okay, it's a polysaccharide, correct? It's a polysaccharide. So that's why our glycogen also, you can see, right? See, our glycogen in our liver is also a form of polysaccharide. So that's why usually our body do tend to store our glucose in this kind of form, which is a polysaccharide form, because the polysaccharide, right, is very inert, and then it's insoluble. So it will not affect our blood osmotic pressure. Okay, so starch is the polysaccharide. So starch is very large and very complex, right? Like a very complex building. So that's why whenever we need to break down starch, for example, we break down the starch, right? Oh, what happened? Oh, sorry. We break down the starch. We add the water to break down the starch. So become what? Become maltose. Starch will be hydrolyzed by water, become maltose. Okay, so maltose is a disaccharide. So it means that this water, right, they will help to break the bonds inside the starch, which is the glycoside bond. Okay, they'll break down the glycoside bond, which is holding the glucose molecule within the starch. So they will break down them and then become maltose, which is become disaccharide. Okay, so if you want to further break down the maltose, become glucose also can. So this is being conducted by what? Amylase. Okay, this is being catalyzed by the amylase enzyme. Amylase enzyme. Okay, so Chloe, what happens if when the starch is being break down by the amylase, they become maltose? So will the iodine solution still able to change their color? Now they become maltose, not starch. Not starch.
No.
Yes, so they will not change their color. So that means that when they become maltose, right, because the iodine solution just remain as brown color, okay? So they will not change color. So that means that iodine solutions, right, they only can change color in the presence of starch only. So you can see, right, how the polymer of glucose are being break down by amylase become the maltose, which is disaccharide. So the structure is totally different. Okay, that's all. Okay, now we talk about lipid. Okay, lipid is quite interesting. Okay, if you want to test this food sample whether they contain lipid or not, so we can just mix them with the ethanol. Okay, ethanol. Ethanol is alcohol, something like alcohol. Okay, so Chloe, may I ask you, ethanol is a polar or is non-polar? Okay, it's something like the non-polar. Okay, it's a non-polar. So that means that this ethanol, right, is a non-polar, which is they can allow the lipids dissolve on it. So, and then you shaken the ethanol, right? And then you added this ethanol with the cold water. You added them with the cold water. So, can the oil dissolve together with water? Oil? Can the oil dissolve together with water?
No.
Yes, no, correct. Because water is polar. Then the oil, right, which is a lipid, is non-polar. So that's why they cannot properly dissolve together each other. So you can see the two separate different layers. But if you added this ethanol, right, so you can allow the lipid dissolve together with ethanol, and then you add with the water. So you can form what? You can form these cloudy emulsions. So if you can see this cloudy emulsion, right, means that there's presence of the lipid inside the food sample. Means there's a lipid inside the food sample. So if they just remain as colorless, right, means that this food sample, they don't have lipid. If there's no lipids, that means that just ethanol, okay, they are shaken with the cold water, right? So you can see totally negative result. So this is, we call it an emulsion test, this one. Okay, because the solution, right, they just appear cloudy, milk-like. Milky color of solution, we call it milky color solution. Because due to the lipid, okay, lipid dissolve with the alcohol and then mix together with water, so they will form this emulsion. Milky emulsions. If you have more lipids concentration inside the food, right, it will become more milky. Okay, which is this one. Okay, cloudy emulsion. This is known as cloudy emulsion. And then I will talk about vitamin C. How are we able to detect the concentration of vitamin C inside the food? Okay, because every food, right, they have a different concentration of vitamin C. So you use this DCPIP solution. DCPIP solution initially is blue in color also. It's blue in color, right? Yeah, it's blue in color. But when they can detect the food sample, okay, they can detect the concentrations, they can detect the vitamin C concentration inside the food sample, right? They can change the blue color, become colorless. Colors means that they become transparent at all. Okay, they become transparent. So it's not blue color, but it's transparent. Transparent color. Okay, this one. Means that if there's a presence of the vitamin C inside the food contents, so it will change the color, become blue, from blue to become colorless or transparent. So the color just look like plain water. It just look colors like water. So if there's no vitamin C inside the food sample, right, it will just remain as blue color. There will be no change in color. Okay, and then usually they use this DCPIP to test for the concentrations of the vitamin C inside the food, and then they can compare which food they have a very high concentrations, which food they have very low concentrations. So they can use this solution DCPIP, and then they can record down they can record down the time change. Means that this food sample, right, require how much for the time to change the solution from blue color become colors. If they require shorter time to change the color, right, means that this solution, this food sample, they have a very high vitamin C concentration. If they require a very long time to change their color, means that they have a very low concentration of vitamin C. It's something like that. Okay, so we already finished the food test. So we just need to remember the color. So we can just realize that a lot of solution, right, initially is blue color, include DCPIP, include this Buret test and Benedict solutions, just accept the iodine. Okay, iodine is yellow brown color. Okay, Chloe, I think you see iodines before, right, in your house.
Yeah.
You know where usually we can use iodine? This one, antiseptic iodine. See, this is the antiseptic iodine. So you can see, right, you can use the iodine solutions to do the starch test. Okay, maybe you can try by yourself. Okay, you can try to drop few on your potato or also on your rice, which is the food that contains starch, and then you can observe any color change. So this is the iodine. Usually this iodine is applied on our wound. Yeah, apply on our wound if we get injured. So whenever you become a vet one of days, okay, you will also use this iodine to do what? To do the request repair the wound for the animals, right? or to clean their wounds, right? So you can see, right, anesthetic iodine in the medicine, right? In the veterinary medicines. You use this solution a lot to clean the wound. Do I have any picture? Wound. Okay, see, those are the okay, they make them become the iodine spray, just spray on the wound. See, iodine to clean the wound, like this. Okay, so if in the small animal, right, for example, dogs or cats, right, it'll be something like this. See? Okay, they're able to clean the wound of the animals by using this antiseptic. It's a very useful antiseptic solution. Okay. So we finished about the food test, right? And then now we talk about nucleic acids, which is the one of the organic compound we haven't discussed yet. What's meant by nucleic acids? How you heard about DNA before, right? DNA.
Yeah.
So what's function of DNA?
Carry genetic information.
Yeah, it's not genetic information. So what is the purpose of this genetic information?
Um.
Okay, this genetic information, right? The purpose is they will like provide guidelines or the instructions for the growth and development of all organisms. And also it helps to do what? It helps to determine the cell characteristic and the functions. For example, Chloe, your hair is black in color, right? And then your iris is brown in color. Okay, you have a brown colored iris. So this is being determined by your genetic. Okay, by the DNA in the body cell. So DNA, right? Okay, DNA is a double-stranded. Actually, we talk about nucleic acids. Nucleic acid we have two types. One is DNA, one is RNA. Okay, so DNA, you know, is double-stranded. It's something like this. You want to draw like this, and then it's no ending. Okay, so this is called double helix DNA. Another one we call is what? RNA. Just like this. This is the RNA, okay? This is the DNA. DNA is double helix. RNA is single strand. It just composed of this single strand polynucleotide. Okay, so what is the difference between DNA and RNA? Okay, DNA is very large and very complex organic compound, but RNA, right, their structure is much more simple compared to DNA, because RNA you only have one strand on it, and then the strand is much more shorter compared to DNA. Okay, so Chloe, I think I have told you a story before, right, which is how our cell produce the DNA. How our cell produce the proteins. So during the process of producing a new protein product, so actually they require to encode, right, copy the genetic information inside this DNA to the RNA. So maybe this region, right, the DNA, they will unwind or uncoil. They will open up. So they will cause this RNA, right, to copy their base sequence, correct?
Yeah.
So you produce what kind of RNA? Messenger RNA. You produce this mRNA, messenger RNA. So this messenger RNA is actually being produced by the enzyme inside the nucleus. We call it what? RNA polymerase. It's a kind of enzyme. This one, RNA polymerase. It's a kind of enzyme help to make this messenger RNA strand. So this messenger RNA strand, right, they can copy the specific genetic information from the specific region of the DNA. So they only copy from here only, here. Okay, they doesn't copy on the whole strand of DNA. No need. They just require to copy a certain region only, because those certain region of the DNA, right, is enough for them to encode to make a protein later. Correct?
Yeah.
Yeah. So the difference between DNA and RNA is just something like this. And then DNA, right? You have the 4 nucleotide base. RNA they also have, but DNA they have this one. They have the A paired with T, C paired with G. But in the RNA they have what? They don't have thymine. They have uracil. Okay, means that you need to pair the A with the U, C with the G. In the RNA they don't have thymine. Instead, they are being replaced with the uracil. Okay, this U means uracil. Okay, yeah. So inside this nucleus of the body cell, they contain a lot of chromosome, and those chromosomes are made from the strand of the DNA. Okay, so you can see, right, a chromosome is made from the long strand of DNA wrapped around the proteins. So become like this. Okay, so this is the DNA. Okay, DNA is the like raw material, so it's something like this. So this is the DNA. Okay, so whenever you want to form a chromosome later, means that this DNA, because DNA is just a strand. Okay, just look like a thread, you know, a long, messy thread. They just coil together. Yeah. So that's why you need to associate this DNA with the proteins, histone. This histone is a type of proteins. So only the DNA they can associate with the histone protein, right? They become chromatins. So now you can see, right, the structure become more stable. You see the structure become more stable. And then you're able to coil this chromatin together, then become the chromosome, which is a butterfly-like shape. Okay, Chloe, how many chromosomes in our body? Okay, the number of chromosomes is about, okay, let me show you. This one. Have you heard of human karyotype? Let me show you. See, this is our human karyotype in every of our body cell. So how they can how they can get this karyotype? Okay, means that they just isolate maybe like your hair cell or the skin cell, right? And then they can do we call it genetic analysis, and then they will come up with this one. Yeah, human karyotype. So it means that in our human body cell, right, we contain about 46 chromosome number. Okay, so the number is about 46. See this one? Okay, 46. So you have about 22, okay? You have about 22 pairs. 22 pairs. 22 pair of chromosome. So what happens is this 22 pair of chromosome, right? If you times 2, multiply by 2, because every chromosome, right, they have about a pair of the chromosome. So you need to times 2, become what? 44. 44 chromosome. Okay, so this 44 chromosome, right, you added up with the 6 chromosome. So when you add up with this 6 chromosome, right, what will happen? They become this one. X and Y, right? XY or XX. Okay, XY will belong to what? Belong to the male. XX will belong to female like us. So 44 plus 2 chromosome at the end become 46. So in human, we have about total number of 46 chromosomes in every of our body cell. Okay, understand?
Yeah.
Yeah. So DNA is a polymer. It's a polymer. RNA is also a polymer. So they are made from the units, which is the nucleotide. So you can see this is the nucleotide. So you have the base, right? 4 type of base together with the deoxyribose and the phosphate group. So this sugar and phosphate group, right, they can join together, right? See, by a bond. Here's a bond. See, here's a bond. Here's a bond. So this bond we know as what? Oh, sorry, this one. This bond we know as the sugar phosphate bond. And then this sugar phosphate bond, like, is a very strong bond. Okay, it's a very strong bond. Strong bond to support the nucleotide structure. Okay, Chloe, later I will share you a video about how a young man, they extract the DNA from the strawberry. Okay, it's very interesting video. So can you just imagine, right? Your DNA. Okay, Chloe, maybe you have a guess. You think, right, your DNA structure is very hard or is very soft? Your DNA structure is hard or is soft?
Soft.
The answer is very hard. Okay. Later I'll share your video. If you have a chance to extract out your DNA from your body cell, you found out your DNA is very strong. Okay, because why? Because they have this one. They have the sugar phosphate bond. Okay, they have this sugar phosphate. Yeah. So they'll make a structure become very strong and very stable. Something, I mean, the concept is just like your cell wall, you know, the plant cell wall, right? Also very rigid, very strong, right? They're able to resist any pressure. Same to the DNA structure also. Okay, can you follow me?
Yeah.
Yes. Yeah. So and then like DNA, right? They can form the base pair. We call it what? Complementary base pair. Is that means that A pair with T, C pair with G. Okay, Chloe, maybe you can wonder what's the reason they can form a base pair? It's because that those base, right, they can join to the opposite strand by the hydrogen bonds. They have this bond. Yeah, they have this bond, join them together. So they can form the pair up A to T, C to G. Okay. And then you can see, right, this sequence like T, G, T, G, T, G, T, also A, C, A, C, A, C, A. Okay, this sequence, right, after they open up, then they will allow the mRNA being synthesized, right? So whenever the mRNA being synthesized, right, so what will happen? Okay, maybe this one open up and then they gone already, okay? Left this one. So maybe call it, we use this strand, right, become our template strand to encode for the proteins. So this is our template strand. So after the mRNA being produced, right, they will become like A to U, C to G, and then A to U. Because mRNA, they don't have the thymine, they only have uracil. Then C to G. Then A to U. C to G. A to U. Okay, so by this sequence, UG, UG, UGU, they can produce another kind of the protein products. But they need to undergo the transition first and the translation. Only they're able to produce a protein product. Okay, so the phosphate and the sugar of the nucleotide, they form the strong backbone of the DNA strands. So, and the base pair of each strand, right, they connect to each other to form the rings of the ladder. Okay, so you can see this is the structure of DNA. You can see, right, this is a nucleotide change like this. And this is another nucleotide change. So that's why DNA means that they have 1,100 unit of nucleotide change. Nucleotide base, they join together to form the polymer. Okay, so this will be the hydrogen bond. See, hydrogen bond. For the DNA, right, you just need to know there's two bonds. First, there will be the hydrogen bond between the base pairing. Second, there will be this one, sugar phosphate bond. I just know this. Okay, sugar phosphate bond. They form a strong back bond. Just these two bonds in the DNA. Sugar phosphate bond is a kind of very strong covalent bond. I'm not sure you heard about this before. Covalent bond. Or you heard about this covalent bond before? Covalent bond.
No.
Okay. You heard in chemistry. Covalent bond. You just start your chemistry, right?
Yeah.
Oh, okay. Your chemistry, you learn about what topic already currently? Atoms, elements, that kind of thing?
We learned about periodic table.
Oh, periodic table. Okay. Yeah. It's an interesting periodic table. Then you learn about what? Group 1, group 2, group 17, I think. Yeah. So it's also related to the biology that you learn. Okay.
Yeah.
So that's why the sequence, okay, which is this one, ACA, right? They will code for the formation of the proteins. Okay. So how we're able to produce the protein products? So that means that DNA, right? They can provide the guidelines first. They can provide a guideline. So the mRNA, they will copy the guideline from the DNA, and then they will go out from the nucleus to the cytoplasm, and then they will meet the ribosome. So after that, ribosome is the chef, okay? It's a cooking chef. So they will just copy the guideline, and then they will prepare the raw material, which is the amino acids. So they can join the amino acids together, and then they will form a polypeptide chain. So the function of the ribosome is what's the function of ribosome? They will produce what?
Produce proteins.
Yes, they will produce the proteins. Yeah. So ribosome, what they do is they will just produce a protein, right, based on the guidelines provided from the mRNA. Because mRNA, they just copy the guideline from the DNA. Yeah. So it means that DNA, they provide the informations, the guidelines to produce the protein product. But the ones who actually produce the protein will be the ribosome, because ribosome is the one who they will join the MSC together. Okay, they will join these three MSC together to become a chain, polypeptide chain. So maybe you can think like, if you don't have the DNA, if you don't have the genetic information from DNA, can the ribosome still produce the proteins without any information from the DNA? Can they?
No.
No, yeah, they cannot. Ribosome, they always need to depend on the DNA. Okay, not DNA, but from the mRNA. Yeah, from the mRNA to produce the proteins. Yeah, which is this one. See? So you can see, right? This is the DNA. Okay, this is the DNA. DNA is very, very large, very, very strong. It's not large and not strong. I mean, very large and very complex, right? Like no ending. You see? Actually, your DNA is no ending. And then inside, there's too much of genetic information. So if you want to make a protein, right, that means that this DNA initially is like this. Okay, so they just open one particular region. Like this, okay? They just open one region. So from this region, right, they're able to produce the mRNA first. Okay, so that's why the mRNA able to copy the genetic information from the DNA, and then they bring out to the ribosome. So ribosome, they'll bind to the mRNA, then they'll read the informations. Just like the cooking chef, right? They actually copy the menu. Like they need to prepare what kind of vegetable, what kind of foods, something like that. So they will depend on the genetic informations on the mRNA, and then they will start to join the amino acids. Okay, so what you can see, because the mRNA, they also have the base pair, right? You see, CGUGA, those are the nucleotide base sequence on the mRNA. So that's why the tRNA, you can see, right, there's another tRNA. The tRNA, they contain these nucleotides, so they can form the complementary base pairing to the RNA. Means the tRNA, they form the base pairing with the RNA to produce the amino acids. to produce a polypeptide chain. Okay, because tRNA, right? tRNA is we call it transfer RNA. Okay, tRNA is a transfer RNA. Transfer RNA, what they do is they will bring the amino acids to the ribosome, so they know it's transfer. Okay, help to transfer the amino acid to the ribosome. Okay, because amino acids, because amino acid is a raw material, okay? It's a raw material to join together to form the polypeptide chain first. After that, they will fold and then they'll become the protein product, the final protein product, which is the, for example, final protein product, okay? So when I talk about the final protein product, we talk about what? What kind of product, which is the proteins? Hormones, right? Hormones. And Chloe, do you know our hair is also made from proteins? Our hair. Okay. The hair protein known as what? I think you heard this term before, right? It's quite I mean, it's quite funny, okay? Keratin. Okay, made out our hair proteins. So keratin is a protein that help to form the hair, nails, skin outer layer. So that's why they have what? They have what? They have the keratin hair therapy. Something like that. Okay, so they believe that this kind of keratin hair therapy or keratin supplement, they can help to make your hair become better, become more elastic. Something like this. See, keratin. Okay, because our hair is made from the keratin proteins. So the keratin protein make our hair become like become increase in their tensile strength. So it means our hair is stretchable, not easily being break up. Okay. Am I speaking too fast for you?
No.
Okay. If I speak too fast, you can let me know. Okay, so clear, Chloe, how the protein able to produce from your body. So this is very important. It's because that your body cell, they just produce a lot of protein product every day. Like your organs produce the enzyme, the hormones, antibody, those kind of protein products. So they all undergo these two ways, okay, which is from the transitions from the DNA first. So the informations from the DNA need to encode into the genes and then transcribe in the messenger RNA. So from the DNA become RNA first. So for the messenger RNA, right, they just carry the information, then they will diffuse out from the nucleus to the cytoplasm, and then they reach the ribosome. So this is translation. Translation happens inside the ribosome. Because ribosome is the one who make the proteins. Translation happens inside the nucleus. Sorry, translation is inside the nucleus. Translation in the ribosome, which is in the cytoplasm. It's not in the nucleus anymore. Okay, then now we talk about the last part, which is the water. Okay, water is a polar. It's a polar, right? So we know as water is a universal solvent. So what I mean by this one, universal solvent. Universal solvent means that water, right, they can allow a lot of substance to dissolve inside. Okay, for example, like the salt particle, sugars, they can dissolve inside the water. So it means that the dissolved substance, they can be easily transported around the organs, like the xylem and the phloem. So it means that the water is usually used for what? Transportations of the nutrient molecule. For example, in our blood also, right? In our blood also contain a lot of the water. So how the blood able to transport their nutrient molecule is they are require the water to help them. And then for this one, digested food molecule in the urinary canal, but need to be moved to the cell all over the body. Okay, so means that whenever we are performing the digestions, we also require the water also. Okay, so if we without the water at this solvent, right, we will unable to allow the food to get into the alimentary canal and then to get into the intestines, the livers, those kind of things. So water is related to the transportations. And then toxic substance like the urea and the substance in excess of requirement, right? They need to be removed away from the body by water. So Chloe, how you able to excrete the waste products? By water, okay? You're able to do excretions, like you are sweating, you sweat a lot, right? When you sweat a lot, you also require the water to help you. Correct?
Yes.
Yeah, because when you sweat a lot, right, you are actually producing the sweat. Sweat is a liquid. So this inside liquid, they contain a lot of mineral salt inside. So means that this water, they can help your body to excrete the excess salt. So water is also important part of cytoplasm. So in our cell, right, we have the cytoplasm. So cytoplasm is inside the contents. They just contain a lot of liquid. So they can allow a lot of metabolic reactions just take place within the cytoplasm because they contain the liquid, which is the water. Okay, Chloe, last question would like to ask you, okay? Water is a polar molecule, right? So Chloe, you tell me, water, they contain any charge inside their molecule? Charge.
No.
That's yes. It's yes.
Oh.
Yeah. Water, they have a charge. That's why water is a polar molecule. You can see, right? The water. So this is also chemistry, okay? Chemistry you learn more. Water is a polar molecule. It's a polar molecule. They can a lot of charge. You can see, right? There's a charge. There will be the negative charge from the oxygen atoms, and then there will be the positive charge from the hydrogen atoms. So that's why water is a polar molecule because they have a charge. Okay, so they know as polar molecule. See. So that's why the water able to allow the sodium, for example, the sodium chloride ion, which is a salt particle, dissolve with it, is because they have charge. Whenever they have charge, right, they can allow the salt attract to them. You can see this is the water, okay? Water from the oxygens, you already know that it's always negative charge. Okay, where is it? So it's always negative charge, right? On the oxygen atom. So the negative charge on the oxygen atom, they will attract to the positive charge on the sodium. Then how about the positive charge on the hydrogen atoms, which is at here? So the positive charge will attract to the negative charge on the chloride ion. So by this way, you can allow the salt particle to dissolve inside the water molecule, which is polar, because they have this polar charge. So polar molecule, they can dissolve with the polar molecule. Polar means that they have a charge. Okay, so Chloe, that's all for today. So far, do you have any question to ask me?
No.
Last question. Sorry, this one. What are the individual units that are linked together to form a DNA polymer? You tell me. So what's a monomer to produce a DNA polymer? DNA, right, is made from what? It's made from the phosphate base and the pentose sugar. So that monomer requires what? Requires the nucleotide, right? Yeah, nucleotide. So nucleotide is a monomer. Okay, it's just like building block, a bridge. Okay, a brick. Later they will become a house, a building. So, which is something like the nucleotide, right? They can join together, so they'll become a DNA polymer. So how about in the protein polymer? Is made from the amino acids. Okay, means that you need to join a lot of amino acids together, become a final protein product.
Yep.
Okay, that's all for today. So we already complete our chapter 4. So I will give you some question to do. You can have a try first. So here's a summary. Okay, so this is we have a 4 organic compound, right? The carbohydrates, proteins, nucleic acid, and the lipid. So this is a monomer. Chloe, you need to memorize this one. Okay, you need to know what's a monomer. What is a monomer? Which contribute become the polymer. So here is a polymer. Here will be the polymer. Okay, polymer include the carbohydrate, proteins, nucleic acid, and the lipid. Monomer is include the basic structure. Basic structure, which is they will join together and then they will become the polymer. So here is a see, you have monosaccharide, amino acid become the proteins. You have the nucleotide become the amino acids. You have the fatty acid glycerol become the lipids. So here's an example. Okay, you can have a read, and then here's the elements. So you can see, right, elements of carbohydrate, they have this one, CHO, 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 CHO. Just the protein, right, you have the extra nitrogen and sulfur. Nucleic acid, you have the extra phosphorus. Okay, and then you should know about functions. That's all. So functions, right, you just need to know that carbohydrate and fats, what they do is they will become the source of energy. Usually the carbohydrate will be the primary source of energy. Okay. So it means that whenever your body cell require energy, so where they can get the energy? Usually they get the energy from the breaking down, which is digestion of carbohydrates first. So if the carbohydrate is being fully depleted, means that there's no more carbohydrates, only the body, they will convert, become the lipids. They will break down the lipids to release the energy. So from the carbohydrates, now they will break down the lipids, become the energy. So lipid is also considered as a source of energy. It's a source of energy. Okay, so that's all for today. So Chloe, if you have any questions, feel free to ask me in the group, okay?
Okay.
Answer you. So I hope you learned something new today. Hope you enjoy the lesson also. Okay, see you in the next class. Bye.
Thank you. Bye.
Bye.